Alright, a very hearty welcome to everyone present in today's session. This session is about the last 10 years of questions that have come from cell cycle and cell division chapter in NEET UG. My name is Ojasvi Singh and I'm going to uh, go through all of those questions with you all and we'll try to solve those questions together. I would personally advise you to pause the screen whenever a question appears before I solve it so that you can give it a shot. You can try to solve it from your side. After you have solved it, you can press play so that you can see whether the answer that you have marked matches with the answer that I'm giving. And I will also explain whatever answer I tick so that it will be useful for you. You'll be able to understand the concepts. And this is something about me. I am currently um, entering my PhD in biotechnology at UNESCO DBT Regional Center for Biotechnology. These are some of the exams that I've cleared. And with that, let's just move directly to the question answer. So the first question says, spindle fibers attached to kinetochores of chromosomes during. So what happens in mitosis? The first stage is, what is the first stage? Prophase, right? Prophase, pro, meta, ana, and telo, right? So in your prophase, basically your chromosomes become visible, the chromatin fibers condense into chromosomes and they become visible. In your metaphase stage, these chromosomes get attached uh, to the spindle fibers at the kinetochores, something like this. So this is your chromosome, this is your centromere, this is your kinetochore, and the spindle fibers attached to the kinetochores. So the answer would be metaphase. Alright? Because in prophase, it's just that the chromosomes become visible. And in anaphase, what happens? The sister chromatids get separated. And in telophase, they reach towards the ends or the poles of the cell. And after telophase occurs cytokinesis, in which these chromosomes, the furrow starts forming so that the division of cytoplasm can occur. And after the division of cytoplasm has happened, each cell will now have the chromosomes also decondense to chromatin fibers, and the nucleus is again reformed around the chromatin fibers. All right, let's move to the next question now. All right, so the next question has two statements. Statement one says chromosomes become gradually visible under light microscope during leptotene. When we are talking about leptotene, it is now talking about meiosis. And meiosis has two stages, meiosis 1 and meiosis 2. Then again, meiosis 1 has a pro, meta, ana, and telo. In prophase 1 of meiosis 1, the stages are again divided into leptotene, zygo, pagaytene, diplotene, and diakinesis. Okay, so over here it says chromosomes become gradually visible under light microscope during leptotene stage. This is true because leptotene is a part of your prophase, and in prophase the chromatin fibers basically condense to form chromosomes. The second statement, so this statement is true. The second statement is the beginning of diplotene stage is recognized by dissolution of synaptonemal complex. So what happens in your prophase 1 of meiosis 1? If I try to draw it, so in leptotene, you basically get your chromosomes. They become visible. In that, okay, I'll just redraw it. So this is what happens in your prophase. In zygotene, Uh, this is leptotene. In zygotine, the homologous chromosomes arrange themselves in the form of bivalents. So this is zygotine, leptozygote. In pakitine, your recombination nodules are formed, and over here your recombination occurs. So this is your leptozygote pakitine. In diplotene, what basically happens is that although they are still arranged in the form of bivalents, but the synaptonomal complex that was formed between uh, the recombining chromosomes, this synaptonomal complex is starts to dissolve. So the beginning of diplotene is synthesized by dissolution of synaptonomal complex is also true. So both the statements are true, right? So this happens in your diplotene, that the synaptonomal complex starts dissolving. And if you want, you can go back to the lecture that I taught in the last video that I have uploaded. Over there also, the statement is clearly written in NCERT that the beginning of diplotene stage is recognized by dissolution of synaptonomal complex. Then again in diakinesis, what happens that this particular part is known as chiasmata. So the chiasmata, the breaking up of chiasmata occurs in diakinesis. So yeah, so both the statements are true in this case. Moving forward, following are the stages of cell division. Choose the correct sequence. So this is actually a very simple question. You just have to remember it because most of the questions are um, going to come from this part. So it's important. So the first thing that happens is G1. In G1 phase is basically the beginning of your, it represents the interphase between it's beginning of interphase between two successive cell divisions. So the and what can I say? So G1 is the first phase. After that comes synthesis phase in which your DNA is synthesized. So G1S, G2 phase in which your proteins and RNA are synthesized. Proteins and RNA. And after that are your stages of mitosis. So all the stages of mitosis, after which occurs cytokinesis. And mitosis again divided into pro, meta, ana, and telo. So let's just arrange it now. So G1, then G2, so E, then A, G1 then synthesis phase, so ECA, and then your karyokinesis, and then last में cytokinesis, ECADB, ECADB. This is your correct answer. Also, since we are on this question, I would like to tell you the significance of G1 phase. So what happens in G1, that the cell basically analyzes whether it has all the necessary requirements to undergo another cell division. So G1 is acting as a checkpoint. G1 has a checkpoint which analyzes whether it has all the things that are necessary for another cell division. If it has everything necessary for another cell division, it will go this route. Otherwise, after G1, the cell undergoes G0. So G0 basically happens when the cell realizes that it does not have sufficient requirements to undergo another cell division, or that it is not necessary for the cell to undergo cell division. If it has everything that is necessary for cell division, then it will proceed via your normal route, that is G1S, G2M, and then cytokinesis, karyokinesis. Now this question is about matching. So, and this is about the prophase 1 of meiosis 1. So the steps, I will repeat again. So the steps are something like, the first stage is leptotene, leptozygo, pakai, diplo, and dia. So in leptotene, the chromosomes become visible. So leptotin chromosomes look like thin threads. Yes. So the beginning of in the beginning of leptotin, the chromosomes look like thin threads. Leptotin, then zygotine is synaptonemal complex formation. Then leptozygopakitine is appearance of recombination nodules. Dikinesis is completion of terminalization of chiasmata. So D3. D3, we have two options for D3. Then pakitine is B4. B4. So this is our correct option. Let's move ahead. The dissolution of synaptonemal complex occurs at. So, if you remember, this is actually the very first question that we had solved. So, the dissolution of the synaptonemal complex begins at diplotin. Terminalization of chiasmata occurs at diakinesis. Okay. Terminalization of chiasmata occurs at diakinesis. Let's move on. Alright, so this question says that the doubling of number of chromosomes can be achieved by disrupting mitotic cell division soon after. So what happens that in your G1, S and G2, then it enters your pro, meta, ana, tedo. So in your G1, the number of chromosomes is, for example, if I talk about the humans, it's 46. And the genetic content is also 46. Then at S phase it's 46, this becomes 92. In your G2 it's 46, this is 92. In your pro phase it's also 46, this is 92. In your meta phase it's 46 and 92. But in your anaphase it becomes 46 and 46 on each side, because in anaphase it starts separating. So 46 plus 46, and in your telophase it becomes 46 and 46 in each cell, right? So if you see, till metaphase you are getting double the number of genetic content, and even though the total number of chromosomes is 46, but those chromosomes look like this. Okay. In your anaphase, the chromosomes will start looking like this because the sister chromatids have separated. So the question, the language of the question is such that it says doubling of the number of chromosomes can be achieved by disrupting mitotic cell division soon after. So you have to stop the mitotic cell division to get double the number of chromosomes. So the correct answer to this would be metaphase. Alright, because after metaphase what happens is that the chromosomes get separated and you get chromatids, and the number of chromatids become 46 and 46 and okay, let me just write over here also 46 plus 46 on each side of the pole. Okay, let's move ahead. During which stages of mitosis and meiosis respectively does the centromere of each chromosome split? So in the previous slide only I just explained that the sister chromatids split at the anaphase stage. So the answer would be anaphase and anaphase 2. To make it confusing, what they could have done is they would have said anaphase and anaphase 1. So in that case, you must remember that at anaphase 1, basically your homologous chromosomes get separated. So the whole chromosome gets separated at anaphase 1, right? However, at anaphase 2, these chromosomes get split and chromatids get separated. And the splitting, basically chromosome split occurs at anaphase 2, and the centromere is split when the chromosome is split. In case of your anaphase 1, what the part that splits is chiasmata. That is this part, when they were attached, that gets separated. However, in your anaphase 2, your centromere gets separated. I'll just write it. In anaphase 2, centromere is split. Let's move ahead. Which one of the following is the quiescent stage? This one is simple. G0 stage is known as the quiescent stage. Select correct sequence of substages. Till this point, it should become very simple and easy for you because we have said it so many times, right? So it starts with lepto, zygo, pagi, diplo, dia. So dikinesis, that is C, should come at last. Okay, three options are C at last. Leptotene, that is so either of these, and then leptozygotine A, TA. So this is the correct answer. The process of appearance of recombination nodules. So basically, they are saying that if this is your pair of homologous chromosomes and over here recombination nodule has formed. So in which stage is the recombination nodule formed? We have already discussed this in the first question itself. The answer would be third stage. So the third stage is leptozygopagi. So pagi. Alright. What happens in the first stage is the chromosomes become visible. In the second stage, this is leptotene. In the second stage, which is zygotine, these homologous chromosomes start to come together as pairs, forming a bivalent, forming an autonomial complex, right? And then leptozygopagitine. In pagitine, what happens is that the chromosomes form this structure which is known as recombination nodule. And around the recombination nodule is formed your chiasmata to give it an environment that supports recombination. Let's move ahead. Which of the following stages of meiosis involves division of centromere? We have already discussed this. It's anaphase 2. Then among eukaryotes, replication takes place in S phase. S phase means synthesis of DNA, and synthesis of DNA basically means replication of DNA from one strand of DNA to become two strands of DNA. This process is known as replication. So replication occurs in the S phase. Let's move ahead. Okay, again it's a match. So let's just go ahead with it. G2 phase means protein synthesis. So B will be 1. B1. Okay, we already got our answer with only just one matching. Let's match the others as well. So quiescent stage is your inactive stage. Then G1 phase is the interval between mitosis and initiation of DNA replication. So A will become equational division. This was an easy question. It has come in 2023, that was last year. Alright, let's move ahead. Select the correct statements. Tetrad formation is seen during leptotene. No. Chromosomes are seen during leptotene. Tetrad formation occurs in zygotene. Let me just write again. Lepto, zygo, pagi, diplo, dia. Okay, so during anaphase, the centromere split and chromatids separate. This is true. So B is true. So only one of these options would be correct. Let's just see D and E because with B only these two options are present. So D says nucleolus, Golgi and ER are reformed during telophase, and E says crossing over takes place between sister chromatids. So the correct answer is B and D. Why is E wrong? So over here E says crossing over takes place between sister chromatids. So in a homologous chromosome, these two chromatids are known as sister chromatids. Okay, but crossing over basically takes place between the non-sister chromatids. Okay, आपस में ये दोनों सिस्टर है और वो दोनों सिस्टर है, but जो recombination होता है, it occurs between the non-sister chromatids, homologous chromosomes. Let's move ahead. So here are two statements. During G0 phase, the cell is metabolically inactive. This is wrong. Cell is metabolically active. Then statement 2 says the centrosome undergoes replication during S phase. This is true. So in S phase, basically your DNA is also replicated and your centrosome is also replicated. Alright. So statement 1 is incorrect, but statement 2 is correct. Let's move ahead. Which stage of meiosis can last for years? Okay, so when I was teaching you the chapter from the NCERT, this statement is clearly written in the NCERT that the stage of meiosis, which can last for months in the oocytes of some vertebrates, is basically a diplotin. Alright, let's move ahead. Identify the correct sequence of events again. So identify the correct sequence of events during prophase 1 of meiosis. So firstly, chromosomes become visible under microscope. So the option has to start from B. We have two options now. Then synapse formation occurs, right? Chromosomes become visible. Then the chromosomes, the homologous chromosomes come close to each other. This is known as synapse. So B, then A. So option D would be your correct option. Then crossing over occurs. Where crossing over occurs, then comes dissolution of synaptonemal complex, then terminalization of chiasmata. Alright, let's move ahead. Bivalent or tetrad formation is a characteristic feature obtained during, so this happens during the second stage, which is leptozygotine. So chiasmata in zygotine stage. Okay, no, chiasmata is not formed in zygotine. Let me just rub this off. Synaptonemal complex is formed in zygotine. Alright, chiasmata is formed in your leptozygopakitine. Let's move ahead. With respect to metaphase, which of the following statements is incorrect? So, chromosomes lie at the equator. This is true. Complete disintegration of nuclear envelope takes place. This is also true. Chromosomes are highly condensed, which is also true. Chromosomes are made up of 4 sister chromatids held together by centromere. Oh, wait a second. Following is incorrect. Chromosomes lie at the equator. Chromosomes are highly condensed. This is true. This is true. Metaphase chromosomes are made up of 4 sister chromatids. Okay. So these three statements are true statements. So, but our question was asking for incorrect statements. So option D would be your answer, because metaphase chromosomes, here it is not saying anything like metaphase 1, metaphase 2, etc. etc. It's not. And then again, it is saying there's 4 sister chromatids. So sister chromatids cannot be 4. Uh, one sister chromatid, the second sister chromatid. So we have only 2 sister chromatids in one chromosome, which is held by one centromere. So over here, the word 4 is actually wrong. It should, if it would have been 2 sister chromatids, then this would have been a true statement. But since it is written in 4, it's a false or incorrect statement. Let's move ahead. Which one of the following never occurs during mitotic cell division? So, spindle fibers attached to kinetochores, movement of centrioles, pairing of homologous chromosomes. In mitosis, there is no homologous chromosomes. In mitosis, they are just chromosomes, right? So, pairing of homologous chromosomes does not occur in mitosis; it occurs in meiosis. Let's move ahead. The appearance of recombination nodules. Again, this is I think the third time this question is repeated. The appearance of recombination nodules occurs during meiosis characterizes sites at which crossing over occurs, and it occurs in leptozygopakitine. Moving ahead, again it's a match. So, and this is actually from the concept is actually from your first lesson, that is on cell, the unit of life. So it says metacentric chromosomes. So metacentric chromosomes are the chromosomes in which the centromere is present exactly at the center, and all of your arms are of equal length. So metacentric would be centromere in the middle forming equal arms. So A will go with 3. A will go with 3. We already have our answer. Alright, this is good when you get the answer so fast. So let's move ahead. Regarding meiosis, which of the statements is incorrect? There are two stages in meiosis, meiosis 1 and 2. DNA replication occurs in. This is an incorrect statement because the correct flow of meiosis is also this. So after S phase, it enters meiosis 1, then it enters meiosis 2. So S phase occurs at the very beginning. Okay. So if it would have been DNA replications occur in S phase of meiosis 1, then it would have been true, but it says that it occurs in the S phase of meiosis 2. So it's an incorrect statement. So let's move ahead. Select the incorrect statement with reference to mitosis. All chromosomes lie at the equator at the metaphase. Spindle fibers attached to centromeres. This is wrong because spindle fibers do not. So see what happens is this is your chromosome, this is your centromere, this is your kinetochore, and spindle fibers are attached to the kinetochore and not the centromere directly. So it should be kinetochore. Spindle fibers attached to the kinetochore of the chromosomes. Let's move ahead. Which of the following stages of meiosis involves division of centromere? This question is also coming the third time or the fourth time, I think. So, division of centromere occurs in anaphase 2. Alright. Um, let's move ahead. Again, it's a match. So, S phase is DNA replication. So, A4. We have two options. Then G2 phase is protein synthesis. So, B1. We got our answer. Quiescent stage is inactive phase and G1 phase is interval between mitosis and initiation of DNA replication. The fruit fly has 8 chromosomes in each cell during interphase of mitosis. Okay, so the total number of chromosomes is 8. During interphase of mitosis, if the number of chromosomes at G1 phase is 8, what would be the number of chromosomes after S phase? So, after S phase, the number of chromosomes still remain 8. However, the genetic content becomes 16. So, the answer to this would be B, that is 8. Okay, so now the question is, which stage of meiotic prophase shows terminalization of chiasmata as its distinctive feature? So, the answer would be diakinesis, right? Because what happens is at your leptozygopakai diplo dia. In your leptotein, the distinctive feature is visible chromosomes appear as thin thread-like structures. Let me just draw it properly, okay? So they start appearing as thin thread-like structures. In your zygotine, the chromosomes come together to form a bivalent and synaptonemal complex is formed. In case of your pakaitin, the distinctive feature is the formation of recombination nodules inside the chiasmata. In your diplotein, the synaptonemal complex starts dissolving, or dissolution of synaptonemal complex would be a feature, right? So, the chiasmata is still not broken. The distinctive feature is dissolution of synaptonemal complex. Just forgive my writing. I'm a bit tired today. So what happens is this is known as your synaptonemal complex. In your diplotein, this synaptonemal complex starts dissolving so that the homologous chromosomes get ready for separation. In your diakinesis, this particular chiasmata is broken. So the chiasmata that was there is broken. So Terminalization of chiasmata is a property of diakinesis. Terminalization of chiasmata. Alright. The centriole undergoes duplication during S phase. S phase involves the duplication of both DNA as well as your centrioles. Let's move ahead. Some dividing cells exit the cell cycle and enter vegetative inactive stage. This is known as quiescent stage. This process occurs at the end of G1 phase. So what happens at the end of G1 phase, the cell can either go and proceed with your cell division, or the cell realizes that it does not want to undergo cell division and it goes to the quiescent stage. Match the following with respect to meiosis. Okay, again a matching. So basically in your leptotene, we don't have leptotene. Leptotene, zygotene. Zygotene synapses. Pachytene crossing over. Diplotene terminalization of synaptonemal complex. Diakinesis, chiasmata. So A will go with A4, B3. A4, B3. We already got our answer. So once again, I think I've made a mistake. Let me just reconfirm it. Zygotene, leptozygotene, synapses, pachytene crossing over, diplotene, diplotene, acha. Okay. Diplotene still has the chiasmata, diakinesis, terminalization of chiasmata. Alright. See, it's not really my fault. I mean, they have just written terminalization. How am I supposed to know? Anyways, let's move forward. So, dissolution of synaptonemal complex occurs during leptozygopachy right? The fourth stage. First, the synaptonemal complex gets dissolved, and after that, the chiasmata is terminated. So, the fourth stage is your diplotene, and it is characterized by the dissolution of synaptonemal complex. Okay, so now the question is identify the correct statements with regard to G1 phase of interphase. So in G1 phase, basically the cell is metabolically active and it is growing, but it has not replicated its DNA yet because the replication of DNA occurs in the S phase. The correct sequence of phases of cell cycle. Oh my god. Are you noticing this? It's like a replication of the same questions again and again in different ways, but the principle, the basics are the same. So correct sequence of phases of cell cycle, so it's G1S, G2M, G1S, G2M. Let's move ahead. Cells in G0 phase, that is your quiescent stage, exit the cell cycle. Alright, let's move ahead. So the stage during which separation of paired homologous chromosome begins is diplotene. It begins at diplotene and ends at diakinesis. Anaphase promoting complex is a protein degrading machinery necessary for proper mitosis of animal cell. If anaphase promoting complex is defective, which of the following is expected to occur? So basically anaphase promoting complex promotes anaphase. That's the basic, like it's just the conversion of the words. So anaphase promoting complex promotes anaphase. So if anaphase promoting complex is defective, the promotion to anaphase would stop and the cell would be stuck at metaphase. So, the basic principle in anaphase is that the chromatids get separated. So if anaphase does not happen, this separation of chromatids will not occur. So chromosomes will not segregate, right? They will not divide, I mean separate. Fragmentation is obviously out of question. Recombination of chromosomes arms will occur. Does not make sense. Chromosomes will not condense. Condensation occurs at the very beginning, that is prophase. So prophase and anaphase, they are quite apart. Which of the following options gives the correct sequence of events during mitosis? So in mitosis, the first thing is condensation, then nuclear membrane disassembly, makes sense, arrangement at equator. So this whole thing is your prophase, pro, meta, ana, telo. Sorry, segregation is also technically under anaphase. So this would be anaphase. So this is your correct answer. Let's move ahead. Okay, during cell growth, DNA synthesis takes place in S phase. Let's move ahead. When cell has stalled, DNA replication fork with checkpoint should be predominantly activated. Now, the language of the question might be a bit intimidating, but the thing is, they're saying DNA replication fork. Cell has stalled DNA replication fork. DNA replication occurs in S phase. So the cell is already in the S phase. Okay, cell is already in S phase, which checkpoint should be predominantly activated. So since G1, so what are basically checkpoints? Like G1S checkpoint will tell the cell that it is okay for the cell to move from G1 to S, from G1 to S. Now this checkpoint has already been crossed because the statement is saying that the cell is already in the S phase. So this is already crossed. Then G2M. So what happens after S phase, the cell would enter G2 and then it will go to M. So from G2 to M. Now if the cell is already stopped at the S phase, it will not be able to enter the G2 phase anyways. So if it is not entering the G2 phase, how will it go to the M phase? So this checkpoint would be activated, G2 to M. Alright, let's move ahead. Match the stages of meiosis in column 1 to the characteristic feature in column 2. So, leptozygopakitine is crossing over. So, A will go with 3. So, we already have the answer because only one option is matching with A equals to 3. Let's move ahead. Spindle fibers attached to kinetochore on the chromosome. Then, in meiosis, crossing over is initiated at pakitine. Which of the following is not a characteristic feature during mitosis in somatic cells? Not a characteristic feature. So, okay, so it is talking about mitosis. So, how will the synapses does not make sense? Synapses occurs in meiosis and not in mitosis. So, this is our answer. Arrange the following events. Okay, again, it's arranging. All right. So, firstly, synapses occurs. After synapses, crossing over will occur. Then terminalization of chiasmata, and then disappearance of nucleolus. So 2 1 2 1 2 1 3 4. This is our answer. All right. All right. So the question says that the somatic cell that has just completed S phase of the cell cycle is compared to the gamete of the same species. So the gamete has haploid number of chromosomes and the DNA content would also be haploid. In case of your somatic cell, it will first enter G1, then S, then G2. So in G1 phase, it is diploid. It is also diploid. It is also diploid. The genetic content is 2N, then it becomes 4N, and then it remains as 4N. And this question is saying that the cell has just completed the S phase. That means the cell is at G2. Okay. So let's compare G2 and our gamete. So the number of chromosomes. So the somatic cell that has just completed the S phase would have twice the number of chromosomes, but 4 times the number of DNA, right? Because the DNA content is 4N over here and it is just N in case of your gamete. Moving ahead, select the correct option. Synapses aligns synapses aligns homologous chromosomes. Synthesis of RNA and protein occurs at G2 phase. So B will go with 3. B3. We have two options now. Then centromeres do not separate, but chromatids move towards opposite. Centromeres do not separate. Okay, this would be anaphase 1. So this is D4. D would go with 4. So we got our answer. So we did not have to solve all of the matchings because with only just 2 we got our answer. However, let's just look at it. A is synapses aligns homologous. Okay, because at leptotene then comes zygotene. In your zygotene, what happens? The homologous chromosomes forms bivalent. This is also known as synapse or synaptonomal complex. And these are your homologous chromosomes. Then we have the action of enzyme recombinase. It also goes with protein. We have done this multiple times. Alright, let's move ahead. So this question says during which phase the amount of DNA in cell remains at 4C if the initial amount is 2C. So let's just try to solve it. We'll start with G1, then S, then G2, then M. So metaphase also again has different stages. Mitosis again has 4 stages: pro, meta, ana, and telo. So in G1, if the amount of DNA is 2C, in S phase it becomes 4C, in G2 it's 4C, in prophase it's 4C, in metaphase also it's 4C, in anaphase it becomes 2C on each of the poles, and at telophase it becomes 2C. So if you're talking about metaphase as a whole, so it also has 4C as well as 2C stages. So metaphase does not have 4C level alone, but the question is saying it remains at 4C, so it will be only G2 because in G2 it remains at 4C. Alright, so this was it from my side. If you have any doubts and any questions, you can reach out to me. My email ID is present in the description, and yeah, that's it from my side. I hope you all learned something new, and yeah, the next chapter, I have not decided which chapter I would upload next, so if you all have any suggestions, please let me know. All right, happy learning.